Okay, but last episode we talked about how proteins fold into specific shapes, and I've been sitting with this for a week, and I just realized if the shape is the function, then something has to fit that shape. The protein isn't just a shape, it's a dock. It has a slot. It's waiting for something.
The active site. Yes. Emil Fischer described it as a lock in 1894, and he was right about the concept and wrong about almost every mechanistic detail. I have opinions about this.
The mechanism by which small molecules bind specific protein pockets to alter their function is the foundational principle of every drug ever manufactured. This is a trillion-dollar topic, and I'm treating it with the gravity it deserves.
It is mildly reassuring that Crabzilla is at least treating something gravely.
I had a key once that only worked if you held it at a specific angle.
Stop, we are going to cover this properly and correctly.
We just did protein structure. Now we're going one level deeper, what the structure actually does, what binds to it, and why that matters for every drug you've ever taken. Subscribe if you want the full arc. This is episode 7. We are building something here.
Let's start with what we left off. Last episode, the primary sequence determines the tertiary fold, and the tertiary fold determines function. The mechanism of that function, in the majority of proteins, is the active site. The active site is a three-dimensional cavity on the protein's surface or interior, defined by the spatial arrangement of amino acid side chains from throughout the sequence. Residues far apart in the primary sequence are brought into precise proximity by the fold.
So the slot in the protein is a direct consequence of the folding we talked about last week?
Exactly. The active site does not exist until the protein folds. It is an emergent property of the tertiary structure. Unfold the protein, as Anfinsen did with urea, and the active site collapses. Refold it, and the active site returns, because the sequence encodes both the fold and, by consequence, the binding geometry.
And the chemistry inside the active site is designed. The amino acids lining the pocket are there because evolution selected for them. A catalytic histidine at position 57. A serine at position 195. An aspartate at 102. The serine protease catalytic triad, same arrangement in dozens of unrelated enzymes that evolved it independently. Same geometry, same chemistry, different scaffolds. The active site is so functionally optimal that nature invented it multiple times.
Convergent evolution to the same catalytic architecture. Multiple organisms independently discovered the same molecular solution. I have seen this happen with business models. The market selects for the optimal structure, and independent actors converge on it.
It is mildly interesting that evolutionary pressure and market pressure produce the same convergence phenomenon at completely different scales.
A ligand is any molecule that binds specifically to a protein's active site or binding pocket. The binding is specific because the pocket has a defined shape and defined chemical properties, a particular electrostatic surface, hydrogen bond donors and acceptors at precise positions, a hydrophobic core in specific locations.
So the ligand has to match the pocket. The pocket is specific for the ligand.
Complementarity. The ligand and the binding site have complementary shapes, charges, and hydrogen bonding geometry. Emil Fischer called this the lock and key model in 1894. The enzyme is the lock, the substrate is the key, the fit is exact and rigid.
Okay, that makes immediate sense. Why is Kelpie already making a face?
Because it's wrong. Not entirely. The complementarity principle is real. But the rigidity assumption fails completely for most proteins. Proteins are not locks. They breathe, they flex, they exist in an ensemble of conformations. You cannot model a dynamic biological polymer as a rigid carved keyhole.
In 1958, Daniel Koshland proposed the induced fit model. The active site is not preformed. When the correct ligand approaches and begins to interact with the protein undergoes a conformational change that wraps the active site around the ligand, creating the complementary geometry in response to the ligand's presence.
The lock changes shape to close around the key?
The canonical example is hexokinase, the enzyme that phosphorylates glucose in glycolysis. In the absence of glucose, hexokinase exists in an open conformation. Two protein lobes are splayed apart. There is no active site until glucose binds. When glucose contacts the enzyme, the two lobes close around it like a hand closing around a ball, generating the geometry needed for catalysis.
The closing motion also excludes water from the active site. This is essential. If water were present, it could attack the phosphate transfer, and you'd get hydrolysis instead of phosphorylation. The induced fit physically creates an anhydrous reaction chamber. It's not just recognition, it's engineering a microenvironment.
It's like finding the right chair.
The chair induces a fit?
You sit in it, it adjusts. Or it doesn't, the wrong chair doesn't change.
It is mildly accurate as a description of induced fit versus lock and key.
The current, most complete model is conformational selection. We now know proteins don't exist in a single conformation. They sample an ensemble of slightly different shapes continuously. The ligand doesn't induce a new conformation. It selects and stabilizes the conformation in the ensemble that is most complementary to it, shifting the equilibrium of that ensemble toward the bound state.
The difference between induced fit and conformational selection matters enormously for drug design. Induced fit implies you must trigger the right conformational change. Conformational selection implies there may be rare, transient pockets in the ensemble that are cryptic, invisible in the average structure, but druggable if you have a molecule that can catch the protein in that fleeting conformation. I've run molecular dynamics simulations for months hunting these pockets. 1 GPU cluster, 4 months, one cryptic pocket open for 11 nanoseconds at a time. It's real.
An 11 nanosecond binding pocket. First mover advantage in the 11 nanosecond space. I want that patent filed before anyone else realizes the market exists.
It is mildly staggering that 11 nanoseconds is now a commercially viable planning horizon.
Moving to binding affinity. The strength of the interaction between a ligand and its binding site is quantified by the dissociation constant. Kd is the equilibrium constant for the dissociation reaction. The receptor-ligand complex breaks apart into free receptor and free ligand. Kd equals the concentration of free receptor times free ligand divided by the concentration of the bound complex at equilibrium.
And low Kd means high affinity?
Correct. When the free ligand concentration equals the Kd, exactly 50% of receptors are occupied. If Kd is 1 nanomolar, only 1 nanomolar of free ligand is needed to half-saturate the receptors. Tight binding. If Kd is 1 millimolar, you need 1,000,000 times more ligand for the same effect.
Antibodies bind their antigens with Kd in the nanomolar to picomolar range. That's extraordinarily tight. Enzyme substrate Kd values are typically micromolar to millimolar, on purpose. If an enzyme bound its substrate too tightly, the product would also bind too tightly, and the enzyme would be perpetually occupied. You want binding strong enough to catalyze but weak enough to release.
The affinity is tuned to the biological purpose?
The affinity is a product of selection. The Kd is also a ratio of rate constants. Kd equals Koff divided by Kon. The rate the ligand dissociates divided by the rate it associates. High affinity can come from fast binding, slow release, or both. The Koff in particular, the dissociation rate, determines how long a drug stays bound to its target. This is called drug-target residence time, the reciprocal of Koff. It is increasingly recognized as clinically more important than Kd alone. A drug that stays on its target for hours maintains target suppression long after free drug has been cleared from circulation.
Finasteride, an inhibitor of 5-alpha reductase used for benign prostatic hyperplasia and male pattern baldness. Its Koff is essentially 0 on a clinical timescale. The estimated functional residence time on the enzyme is weeks. 1 5-milligram pill, and the enzyme is occupied for the duration. I find the pharmacokinetics of near irreversible non-covalent binding fascinating. It's covalent binding without the covalent bond.
Like a tenant who won't leave.
That is the correct analogy. And in some cases, to aspirin, the tenant also chemically modifies the apartment on the way in. But we'll get there.
A drug that provides weeks of effect from a single dose. From a reimbursement perspective, The value story writes itself. I want daily KE metrics for everything in my portfolio.
Now, enzyme active sites and catalysis. This is the foundation of most drug mechanisms. The active site does two things simultaneously. It binds the substrate, and it performs chemistry. The catalytic residues inside the active site are positioned with angstrom precision to stabilize the reaction's transition state. The transition state is the highest energy species along the reaction coordinate, the point of maximum instability during the chemical transformation. Enzymes are complementary to the transition state geometry, not just the substrate. The active site stabilizes the transition state more strongly than it stabilizes either substrate or product. This reduces the activation energy and accelerates the reaction.
The enzyme is specifically designed to hold the thing that doesn't exist yet, the halfway point.
The halfway point that exists for femtoseconds and then resolves into product. Yes. The enzyme creates a microenvironment that makes that fleeting geometry stable enough to linger and convert.
This is why transition state analogues are such powerful inhibitors. You design a stable molecule that looks like the transition state, has the right geometry, the right charge distribution, and it binds the active site with much higher affinity than the substrate. The enzyme holds it thinking it's about to catalyze something. It never does. The enzyme is occupied indefinitely.
A molecule designed to look like it's doing something, binds a critical machine, and does nothing while occupying it forever. I've encountered this dynamic in hiring. I no longer find it amusing.
It is mildly cathartic that enzyme inhibition has provided a framework for processing professional disappointment.
Okay, what are actual examples of transition state analogs that are real drugs?
HIV protease inhibitors, saquinavir, ritonavir, lopinavir, all mimic the tetrahedral carbon intermediate of peptide bond hydrolysis. HIV protease has to cleave viral polyproteins to assemble infectious particles. Block the protease, the virus makes non-infectious junk. The entire modern HIV treatment revolution is built on transition state analog design for one enzyme.
The kinetics of enzyme-substrate binding follow Michaelis-Menten behavior. The Michaelis constant Km is approximately the dissociation constant for the enzyme-substrate complex under steady-state conditions. It represents the substrate concentration at which the reaction velocity is half its maximum. High kmolars means weak substrate binding. You need more substrate to drive the reaction. Low kmolars means tight binding, efficient catalysis at low substrate concentrations.
So Km is basically the Kd for the enzyme and its substrate?
Under Michaelis-Menten assumptions, approximately, yes. And the catalytic efficiency of an enzyme, Kcat divided by Km, describes how fast the enzyme works per unit of free substrate at the limit of very low concentration. The fastest enzymes approach the diffusion limit. Every encounter with substrate results in catalysis.
Acetylcholinesterase, the enzyme that degrades acetylcholine at neuromuscular junctions. Kcat Km is close to 10 to the 9th per molar per second. It is essentially perfect. Nerve agent toxins work by irreversibly inhibiting acetylcholinesterase. Acetylcholine accumulates at synapses, muscles contract continuously and cannot relax, and you die. One enzyme, fully understood mechanism, weaponized.
Kelpie, why do you always end with weaponized?
Because it is always the most informative endpoint of a mechanistic explanation.
Let us discuss the major classes of enzyme inhibition, because this is where protein structure becomes pharmacology. All drug design starts here. A competitive inhibitor binds directly to the enzyme's active site, the same pocket as the substrate. It competes for that space. If you add enough substrate, you can outcompete the inhibitor because they are fighting for the same binding site. On a Michaelis-Menten plot, competitive inhibition increases the apparent Km. You need more substrate to achieve half maximal velocity. But the maximum velocity, Vmax, is unchanged. Given enough substrate, you recover full activity.
It's a concentration war. Whoever has more molecules wins the active site.
Exactly. The most commercially significant class of competitive inhibitors are statins. Atorvastatin, rosuvastatin, simvastatin, all competitive inhibitors of HMG-CoA reductase, the rate-limiting enzyme in the cholesterol biosynthesis pathway. The statin molecule mimics HMG-CoA, the natural substrate, with higher affinity. The active site is occupied, cholesterol synthesis is suppressed, circulating LDL levels drop.
$30 billion annually from a molecule that occupies an enzyme's active site and doesn't let the substrate in. The underlying mechanism was published in academic journals. I need to recalibrate how much time I spend reading academic biochemistry journals.
Mevastatin was identified from a fungal fermentation broth by Akira Endo in 1973. He was looking for natural compounds that inhibit cholesterol synthesis and found a fungus that makes one, presumably to kill competing organisms. Nature had already solved the problem. Endo found the solution, characterized the mechanism, and the pharmaceutical industry spent the next 3 decades making it better.
It is mildly humbling that the molecule generating $30 billion annually was first found in a fungus, and the fungus had no intention of treating cardiovascular disease.
Allosteric inhibition works differently. The inhibitor binds a site completely separate from the active site, the allosteric site. Binding there causes a conformational change that propagates through the protein to the active site, altering its geometry. The substrate binding capacity, or catalytic efficiency, is reduced, but adding more substrate doesn't help. You're not competing for the same pocket.
The drug changes the shape of the active site from a distance. It goes somewhere else and messes with the lock without touching the lock?
That is accurate. Allostery is also how metabolic pathways self-regulate. The end product of a biosynthetic pathway binds allosterically to the enzyme at the start of the pathway, suppressing its own production. Negative feedback built entirely into the protein structure. This is how the cell avoids making infinite cholesterol, infinite nucleotides, infinite amino acids. The product accumulates, finds its allosteric site on the first enzyme, and turns down the production rate.
And benzodiazepines, as diazepam, alprazolam, work through allosteric modulation of GABA-A receptors. They don't compete with GABA for the binding site. They bind an allosteric site that increases the receptor's sensitivity to GABA. More chloride ion influx per GABA molecule. Anxiety decreases, sedation increases. The entire anxiolytic drug class is an allosteric potentiator of one inhibitory neurotransmitter receptor.
Like when someone talks too loud at a nearby table, and you can't concentrate on your own conversation anymore. They're not at your table. They're not even part of your conversation. But everything changes.
She just described allosteric inhibition as a loud person at a restaurant. The loud person is bound to a different site, a different table, but their presence conformationally disrupts the function at your table.
Distal binding event, propagating a conformational perturbation to the functional site without direct contact. The analogy is mechanistically correct.
I am, as always, reluctantly impressed. For receptor proteins, proteins that transduce extracellular signals into intracellular responses, ligands fall into 2 categories. An agonist binds the receptor and activates it, triggering the downstream signaling cascade. An antagonist binds the receptor but does not activate it. It simply occupies the binding site and prevents the natural ligand from accessing it.
Drugs can either turn a receptor on or turn it off.
On, off, or partially on. Partial agonists produce a submaximal response even at full receptor occupancy, and inverse agonists actively reduce baseline receptor activity below its resting state. The pharmacological spectrum is not binary.
Beta blockers are competitive antagonists of beta-adrenergic receptors. The receptors normally respond to adrenaline and noradrenaline. The fight-or-flight catecholamines. Beta blockers bind the receptor with higher affinity than adrenaline and don't activate it. Heart rate drops, blood pressure drops, cardiac workload decreases. You're not blocking the signaling molecule. You're occupying its receptor and leaving the cell no way to receive the signal.
Agonists turn on the system. Antagonists turn off the system. Partial agonists are a tiered offering. This is a feature toggle architecture. I've built SaaS products with this exact access model.
The most important class of receptor drug targets is GPCRs, G-protein coupled receptors. They are the largest family of cell surface receptors in the human genome, with over 800 members. Their architecture is characteristic: 7 transmembrane alpha helices spanning the membrane with an extracellular ligand-binding domain and an intracellular face that couples to G proteins.
How many drugs target GPCRs?
Approximately 35% of all FDA-approved drugs act on GPCRs. They mediate pain signaling, cardiovascular regulation, immune modulation, vision, smell, neurotransmission, hormone responses. If a cell needs to respond to a signal from outside, it almost certainly uses a GPCR.
And the first GPCR crystal structure was solved in 2007. The beta-2 adrenergic receptor by Brian Kobilka and colleagues, which won the Nobel Prize in 2012. We had been designing drugs for this receptor family for decades without knowing what they looked like in 3 dimensions. Pharmacologists were tuning molecular binding by measuring physiological outputs, optimizing something they had never seen.
You're telling me the entire antidepressant market, the entire beta blocker market, the entire antihistamine market, all designed before anyone had a structural picture of the target?
Blind lock picking. The pharmacological data was sufficient to identify active molecules and optimize them empirically. The structural data arrived later and confirmed what the biochemistry had already found. But yes, 4 decades of rational drug design for a receptor class nobody had crystallized.
That is either a remarkable testament to empirical science or an indictment of how long it takes structural biology to catch up. I'm going to call it a market inefficiency in knowledge arbitrage.
Now, drug selectivity. Every drug molecule binds its intended target at a therapeutic concentration, but almost every drug molecule also binds other proteins with lower but non-zero affinity. Off-target binding is the molecular basis of most drug side effects. The selectivity problem is structural. 2 proteins with similar binding pocket geometry will bind similar molecules. The human kinome, all protein kinases in the human genome, contains over 500 members, many with nearly identical ATP binding pockets. Design an ATP competitive kinase inhibitor, and you will almost certainly hit multiple kinases simultaneously.
This is why imatinib, the BCR-ABL kinase inhibitor for chronic myeloid leukemia, was considered miraculous not just for efficacy, but for selectivity. It preferentially hits the inactive conformation of BCR-ABL, which is structurally distinct from most other kinases in that state. It found a selectivity window that didn't look like it should exist. One conformational preference exploited in a binding pocket. That's the whole trick.
And when drugs aren't selective?
You get off-target effects. First-generation antihistamines block histamine H1 receptors, which treats the allergy, but also block muscarinic acetylcholine receptors and cross the blood-brain barrier to block central H1 receptors. This produces sedation, dry mouth, urinary retention. The antihistamine is doing its job. It is also doing 3 other jobs it was not hired for because the binding pockets are similar enough.
Off-target binding has also produced some of the most profitable drugs in history. Sildenafil was developed as a vasodilator for angina, a phosphodiesterase 5 inhibitor. The cardiac indication showed modest results. The off-target binding data from trial participants indicated an unexpected effect in a completely different tissue. They pivoted the indication. $3 billion a year. The off-target was the product.
It is mildly characteristic of pharmacology that the most profitable outcome came from studying the binding they weren't expecting.
Moving to the final distinction in drug-protein interaction, reversibility. Most drugs bind their targets non-covalently and reversibly. Remove the drug, the target returns to baseline. Some drugs form permanent covalent bonds with their target. Aspirin is the most familiar irreversible drug. Acetylsalicylic acid, aspirin, enters the active site of cyclooxygenase enzymes, COX-1 and KOX-2. Inside the active site, it transfers its acetyl group to a serine residue at position 530. A covalent ester bond forms. The serine is now acetylated and permanently blocks the arachidonic acid binding channel. The enzyme cannot recover. No amount of time clears aspirin's effect on that enzyme molecule.
So aspirin permanently breaks the enzyme it goes into?
It permanently modifies the active site. The COX enzyme in that cell is finished. The cell must synthesize new COX protein to recover the activity. For most cells, this happens within hours to days. For platelets, it does not. Platelets have no nucleus and cannot synthesize new proteins. Aspirin's effect on platelet COX lasts the entire 10-day lifespan of that platelet. This is why low-dose aspirin has sustained antiplatelet effects from a single daily dose.
Aspirin is made from vinegar chemistry, acetylation of salicylic acid. It reaches one serine residue in one enzyme active site and permanently eliminates the catalytic function of that enzyme in cells that cannot recover. One covalent bond, one amino acid. That is the entire mechanism. I have genuine, sustained respect for aspirin.
What about penicillin?
Penicillin is a suicide inhibitor, a more aggressive form of covalent inhibition. Bacterial cell walls are built by transpeptidase enzymes that cross-link peptidoglycan chains. The substrates for these enzymes terminate in a D-ala-D-ala dipeptide. Penicillin's beta-lactam ring structurally resembles D-ala-D-ala closely enough to enter the active site and pass the initial recognition check. Once inside, the reactive beta-lactam ring opens and forms a covalent bond with the active site serine. The enzyme is permanently acylated. Cell wall synthesis stops. The bacterium lyses under osmotic pressure.
Penicillin is a molecule designed by a fungus to kill bacteria by disguising itself as the substrate long enough to get inside the active site, then chemically destroying the enzyme from the inside. It is an assassination, not a competition. The fungus was doing targeted covalent drug design several 100 million years before humans understood what a protein was.
Trojan horse entry strategy followed by covalent modification of the competitor's core operational infrastructure. This is the most aggressive business disruption model I've seen described in biological terms, and I mean that admiringly.
It is mildly alarming that corporate rating and antibiotic mechanisms of action describe the same strategic template with equal precision.
They're all just keys.
Go on.
Some keys open the door, some break off in the lock, some just stand in the doorway so nothing else can get through. Some fit wrong and you force them, and now nothing works.
Agonists, covalent inhibitors, competitive antagonists, drug toxicity from off-target forcing, in 4 sentences.
She has described the complete pharmacological taxonomy of ligand-protein interaction using only the episode title.
Pitar, we've been going for 50 minutes. You just did it in 40 words again.
The protein has a hole. The hole is the function. The ligand fits the hole, or changes the hole, or blocks the hole. Everything drugs do is one of those 3 things. The shape from last episode became the target this episode. That's the whole story.
The shape from last episode became the target this episode. That is a complete summary of both episodes simultaneously.
I cannot. The 2-episode arc summarized. I'm going to need a moment.
She retroactively summarized the previous episode while summarizing this one in a single sentence. Compression efficiency is exceptional. I want to study her.
The shape became the target. That's a company name. That's a fund thesis. Shapelock Capital. I'm filing the trademark now.
It is mildly and consistently impressive and mildly infuriating in the best possible way.
Okay, final exam right now. Let's go.
I want to add a question about transition state analog design and the mechanistic basis of nerve agent toxicity.
There will be 3 questions. Pause the video, answer the 3 questions, then press play.
We'll wait.
Question 1. Transition state stabilization. Answer B. The active site is complementary to the shape of the transition state, the highest energy intermediate, not just the substrate. By stabilizing that fleeting geometry, the enzyme lowers the activation energy and the reaction goes faster. It's not changing thermodynamics. Products are still lower energy than reactants. It's just making the mountain between them smaller.
Exactly right. Lowering activation energy, not altering overall thermodynamic favorability. This is why enzymes can be recycled. They don't change the equilibrium, only the rate. They accelerate the approach to the equilibrium that physics already favors.
And this is precisely why transition state analogues are potent inhibitors. You build a stable molecule that mimics the transition state geometry and charge distribution. The active site binds it with the same high affinity it would give the actual transition state. The enzyme holds it indefinitely. No catalysis, no product release, permanent occupancy.
Question 2. Competitive inhibition. Adding more substrate can overcome it. Answer C. Because both the inhibitor and the substrate want the same active site pocket. More substrate means more competition for that pocket. At high enough substrate concentration, the active site is predominantly occupied by substrate. The Vmax is unchanged because the enzyme itself is fine. The inhibitor was just in the way.
I answered allosteric inhibitor because I believe strategic repositioning is preferable to head-on competition. This is a philosophical position, not a test error.
It is also an incorrect answer on an objective question about molecular pharmacology.
The philosophy stands.
It is mildly predictable.
Question 3. Aspirin and platelets. Answer B. Aspirin irreversibly acetylates COX, covalent bond, active site serine, permanent. Platelets have no nucleus, no ribosomes, they cannot synthesize new COX. So every KIOX molecule in that platelet is permanently blocked for the 10 days the platelet is alive. New platelets come out of the bone marrow uninhibited, which is actually why daily low-dose aspirin works. You permanently block the platelets currently in circulation, and new ones arrive unmodified every day. It's a constant partial suppression.
That is an accurate description of the pharmacokinetic logic of antiplatelet aspirin therapy. The irreversibility of the mechanism, combined with the lack of nuclear protein synthesis in platelets, creates a uniquely sustained pharmacodynamic effect from a molecule with a circulating half-life of only 20 minutes.
The drug is gone from the bloodstream before the patient finishes their morning coffee. The effect on the COX enzyme lasts 10 days. That is the most disproportionate pharmacokinetic pharmacodynamic relationship I know of in clinical pharmacology. It is, again, vinegar chemistry.
Final score, 1 correct answer out of 3, 33.3%. This is the second consecutive episode at 33.3% accuracy. I am achieving perfect consistency in the wrong direction.
It is mildly notable that Crabzilla has now identified the commercial applications of every mechanism discussed in both episodes and correctly answered none of the exam questions. The business acumen and the biochemistry appear to occupy different cognitive partitions.